to my second lecture. Okay, now I need to click something here. Good. Probably not 100% sure. Maybe I need to, no, it doesn't ask me here to confirm. But yeah, I think it worked.
But
what?
It's the setup you're using last week, right? So last week.
Same setup.
So,
yeah. Good. All right. So yes, welcome to my lecture. Today we talk about learning and plasticity, which is, if you ask me personally, one of the most important topics, and not only in neuroscience, also in today's machine learning. It plays a huge role. When we go to the content of the lecture, these are kind of three textbooks that you mostly find online if you actually search. If you want to read up on these things, there's the Kandel, very famous Eric Kandel, Nobel Prize winner, wrote this book, Principles of Neuroscience. Then we have the Squire, the 4th edition, and the 3rd edition you find online for free as a PDF if you Google it a little bit, and it's almost the same. So pictures are not as nice, but you know, it's the same content. And then also, I think online you can find this theoretical neuroscience book from Diane Abbott, really a classic, you know, talking about also things from last time. Some people ask for, you know, more literature with respect to tempo and rate coding. You should be able to find this in here. Good. All right, so content of the lecture today, I would really like to take some time and motivate the topic. Why do we need plasticity and learning? And also, why might it be interesting to look or understand biology when you are a machine learner? Then we'll define plasticity and learning, at least for the brain mostly. For computer science, it's quite simple. Then we go through the relevant timescales for learning. And this is also crucial because in the brain, you'll see that there's a couple different mechanisms and plasticity mechanisms that act on completely different timescales. Then we'll go through substrates of neuroplasticity. And here we have basically 3 levels. And the 3 levels are, first, we look at brain areas, right? What happens, for example, if brain areas such as the hippocampus does not work anymore? What types of memories can people not form anymore? Then from there we go to the cellular plasticity, right? What happens when we learn new things on the cellular level? We talk about the perceptron. That is kind of, you know, a computational idea of how plasticity works and how cells integrate information. And then we'll go from there to synaptic plasticity, the Hebbian synapse, and then also go a little bit into just briefly into the molecular underpinnings of synaptic plasticity. Good. So I would like to start with this picture here. This is the C. elegans worm. And I wanted to, you know, shoot a question at you, which is basically this worm in principle does fine, right? It exists since, you know, many thousand years on this earth. In this worm, it has about 108 neurons in its head, and all of these neurons are kind of more or less the connections between these neurons and the genetic background is all encoded in the genome, right? It has about 7,000 synapses, 302 neurons. There's other neurons in its body which control digestion. So, but everything is encoded in the genome, right? And so these things, you know, they do learn almost nothing. So they come to this world and they seem to be fine, right? They exert certain, you know, behaviors, not too complex. So why is this not a strategy for the brain, right? So for example, for us, right? Why can we not just, you know, do that? It wouldn't be awesome, right? Everything I learned, I'll just over time integrate, I store in my genome, and then somehow I, you know, pass it on to my next generation. Any ideas? Please unmute yourself if you have an idea.
Maybe to adapt to changes in the environment.
Yeah, good point. Yeah, that's one thing. But let's ask the genome question first, right? And I'll do a little over the thumb math calculation. Should come up in a second. No, it's always slow here. Okay, good. So when we look at the human brain, so the question is first, right, is it even possible to do that, right? Can we store all the connections in the brain on the human genome, right? That would be awesome. So the human brain has 10 to 11 neurons, right? Every neuron has, on the order couple of 100 to 10,000 of connections. We say like 1,000, synapses per neuron. And then the synapses, of course, not binary, so we have a certain synapse bit depth, right? So here we assume 35 bits or something, which is probably relatively conservative. Then we can, calculate to sum this up, and we would, basically go to half a petabyte or something storage that we would need to store everything that we have learned on a hard drive. Okay, and this is still conservative because there's other types of signaling in the brain, right, apart from synaptic signaling. So when we look, however, at the genome, right, then we'll find that the genome only has 1 gigabyte of information, right, and probably only 20% of this genome encodes our human brain. So we have, maybe even less, right, 200 megabyte. Maybe this is discussed, right? Many of this data is not even encoding anything. So we might be left with a relatively small data space that we can actually use to store information that we pass on to somehow our next generation, right? So I personally see this as a biological bottleneck, right? This doesn't exist from machines and robots. You would just download everything on a big hard drive and pass it on to the next one. But this forces us, right, to be extremely good learners, right? And this also forces us to have really sophisticated mechanisms in the brain that allow for a very, very effective learning. Yes, good. Okay, this is one point that I wanted to make. And then, of course, I'll ask this question here, and I'll show you this movie. I can talk about running. This is I was fascinated by this recently. So, think of a car, like building a car. This is a car manufacturing don't tell you what it's. But when you think about building a car, it's a really complex thing, where I would argue you need some type of intelligence to do these robots. And then somehow I can ask the question, are these robots as smart? And you can watch this movie for like half an hour and you won't see any human in it. Yeah.
Can you repeat everything you said during the video? I think your voice.
Okay, good. Good. Yes. So this video, right? Obviously, building a car is a very complex thing. Where I could argue you need some intelligence to build this car, right? You can just do this. And these robots somehow they do something very, very, very complex, right? And but the question is somehow are they really intelligent, right? And of course. Something that these robots cannot do, right? Think about if one screw drops on the ground, or if one car is positioned a centimeter off, right? Then they completely screw up, right? And this is exactly what some of you said, a minute ago. When the condition is changing, when the environment is changing, right, you need learning and plasticity to adapt. And this is where animals, that's why I showed that picture from this little bagger here, animals and humans are extremely good in adapting to changing conditions, right, to relearning things. So this guy here, where's the water puddle? Where does he find nuts in the winter and the summer? His conditions are never stereotypic and are always changing, right? And that's the second thing why we need plasticity and learning. So the idea here is, and I have discussed this already with you, that basically learning is a very strong, seem to well designed evolutionary survival strategy. If we look at this plot on the right, you could plot performance on the left. So this is, like a evolutionary fitness or something, right? How well do you do in this environment? And the blue curve would be our C. elegans, right? We would have a very good performance, let's say, from the beginning, but then there's almost no learning because we have no plasticity, right? At the same time, we need to store everything on the genome, right? Which is possible if you're kind of, a small organism. If you look at the red curve, the strategy is different. You say, well, I want to, at the end, perform much better, right? But I can't store too much on my genome. So I start with somehow, at a point where I don't do much, right? I have some very basic rules that allow me to learn very quickly, very efficiently, right? And then I grow my behavior performance over time so that I get much, much better. And therefore, I need a strong plasticity. Of course, I also need some type of nurturing usually, right? Which we see in mammals happening. Okay, and I encourage you to read this this review here from Anthony Zeta that's kind of exactly discussing this issue. Okay, good. So kind of last couple of slides and then I stop motivating learning. My kind of, all right, good. Let me go back here. It doesn't really. Okay, yes. So everything that I think we do, right? Think about it. Running, singing, playing musical instruments, writing, everything is learned, right? We cannot do almost anything that we haven't learned, right? So I think if we think of intelligence and intelligent behavior, then it's clear that learning is very, very crucial for all of this. Good. This is just the last slide here. This is just to show you, this robotic arms, what kind of machine learning can do at the moment, right? To just make the point that humans are still much, much better in learning than any of these machines. So here, Google trained these arms on millions and millions and millions of bin picking, right? Now they can do this, more or less good, right? And then you have on the other hand, children that maybe have, done a couple of, let's say, a couple of 100 trials and are able to do that kind of with ease. So to say, this is just to motivate the point that I think we can still learn a lot from biology and then possibly also improve machine learning, reinforcement learning, and other algorithms in a variety of tasks. Okay, good. So enough motivation. Let's now go to the definition of plasticity and learning. And just to when you come from a neuroscience perspective, what you'll find is that these definitions often come from the field of cognitive neuroscience and psychology. I'll comment on this in a few slides. Okay, good. So learning and plasticity. Okay, good. Come on. There we go. Okay, good. So learning is the acquisition or storage of knowledge, right, through experience. In machine learning, you would say training, right? And you have training examples. Here is learning and experience. Memory is the stored information that you can recall, right? Learning usually results in memory, which in the normal case has a certain outcome, right? It changes behavior. We can do something better. We can ride a bike. But be careful, not all learning, can directly be observed at the behavior level, right? If you just look at, the bus every morning at 8 and you notice it's blue and you learn things, right? Might not change your behavior because it's irrelevant, right? It might change your behavior exactly at a time where somebody asks you which color has the bus or where you need to take the bus. Good. Okay. Then plasticity is basically the biological implementation of learning, right? It allows us to form memory. Okay, and this is a classical learning curve. This is also from the Kandel book, where people kind of were asked to draw the star, right, within these boundaries. They repeat it, they repeat it, they repeat it, and then they get better at it. And on the bottom, you basically see the, the first, second, and third day over trials, the performance over trials. And what you see is, and this is very, very similar to, machine learning. When you take classifiers, the deep nets, you see the, the decrease in errors with learning, right, with, data samples. Okay, good. Just to put kind of, learning in the context of, okay. Just to make the difference clear in computer science, right? When we talk about learning, we talk about supervised learning, we talk about unsupervised learning, right? Where you don't have labels. We talk about reinforcement learning, where you're an agent and you explore and you learn, right? And this is very different in neuroscience. If you are in neuroscience, then you'll kind of find these types of expressions. For example, Pavlovian conditioning is a certain type of learning that's often discussed in neuroscience. What's Pavlovian conditioning, right? Very simple. The mouse, the animal, what you see here on the right, upper right, sits in a little box. It gets a tone, and then either it gets a reward or it gets a little shock, and then it learns just to associate these two cues, right? Instrumental conditioning is the extended version of this. What you also see on the bottom right, there is an animal, and this animal somehow gets a cue, can press a lever, so it has actually to do an action, right? It goes more towards reinforcement learning, and then it gets a reward, right? So it has to do something. Okay, then you might read reward aversive learning, right? That's kind of, it's not orthogonal to these other two, right? Both of them can be either done with a reward or with an aversive, with a painful stimulus, right? Just to make a note, mice, we work, for example, with mice and rats, they wouldn't do anything. They wouldn't learn anything if you don't reward them or if you don't, provide some aversive stimuli, right? So for us, actually the only, usually we give a reward, right? The only way to make them learn anything. Then there's social learning, right? I think about maternal learning, where young mice learn from their parents. Perceptual learning and motor learning are also orthogonal to these upper points. Perceptual learning is really, you learn, for example, discriminating certain stimuli, right? You learn to be more sensitive to certain stimuli. And then you have motor learning, where you, for example, the mouse learns to walk very fast across a ladder or something, right? And after it's done this 100 times, it can do this better. Good. So what types of memory do we have? And this is a classification from psychology, right? And I would like to kind of make the disclaimer here that this is what psychology comes up with, right? On a conceptual level. That doesn't mean that the brain somehow really has, the same. The brain might use, all brain areas to implement these memories, right? So we don't know if on the neural substrates this kind of classification actually makes sense. Good. Our memory stores personal experiences, emotions, preferences, and dislikes, right? Which food do you like? Motor skills, obviously riding a bike, something like this, right? World knowledge, this is factual knowledge. I'll have an example on this. Language, for example, is all learned, right? Cognitive memories, you are able to recognize somebody or some object. Then, of course, very important, we'll talk about this today a lot, spatial memories, right? You remember in which room, in which place you are, or how to get from A to B. Good. And psychology also makes this point that kind of we, right, are fundamentally derived from our learning experience. That's what basically makes us. Good. And then psychology further classifies memories into two forms. One is explicit declarative memories, and these are facts and events. This is basically something that I can ask you. You could give me an answer, right? If I ask you, what's the number pi or something, you could tell it to me. And then there's implicit memories or non-declarative, such as priming, procedural skills, right? How do you ride a bike or things like this, right? You just can learn it, but you can't maybe tell me how to hold the balance on a bike or something, right? Then there's associative learning, for example, emotional associative learning, your emotional response to a certain person, right, is implicit memory. Then you have non-associative learning, you have some, habituation and sensitization. If, for example, right, something hits you on the arm, right, and maybe it hurts, then you get sensitized to this, right? This is something that's also implicit. Okay, good. These are two examples for explicit or declarative memories. Basically, I can ask you who won the world championship last time, or facts, right? What's the number P? And then this is an example for an implicit memory. So I would just show you these words here, right? Then I would blend them out, and then I would, during recall, I would somehow ask you a question, something like this. Okay, good. Coming. We are meeting two, and then I would ask you to fill this out, right? And because you just remember certain things, right, you would somehow fill it in the right context. Okay, good. All right, so I would like to talk for a moment now on the relevant scales of audio. Okay, the relevant tempo scales for learning. And they're basically, you have kind of, I think, 4 categories. You have the very short term, right? And here we're in the order of somehow, okay, good. Okay, good. And here we are basically in the order of seconds, right? And we're somewhat short term memory, right? And this is seconds. Seconds. Okay, good. 2, we basically, this is already called long-term memory, so LTM. And this is where you may have heard LTD or LTP, right? This is all here. Good. And this is on the order of minutes. Then we have basically I would call this long-term or very long-term memory. This is in the order of hours to days. And then the last category, what we have is 4, where we have the long lasting. Good. Oh dear. Good. Good. Memory, where we have months to a lifetime. Okay, good. So why does it make sense to have these different types of short-term memories, or how do we classify them? You see that they're characterized by different mechanisms, right? And we'll go through these later. Short-term memory, for example, is usually, built up of calcium in the synapse, right? Long-term memory is really starting with protein expression. Very long-term memory, you see that somehow new synapses form, right? And very long-lasting memory, you see complete structural changes. There's new connections from one brain area to the other, for example. Good. And this is an experiment that basically shows you that somehow also different brain areas are related to these different memory timescales, right? So this paper here I brought up basically saying that the hippocampus is involved mainly in short to long-term memory processing, but not super long, right? I can explain this briefly. So here there was a group of rodents that learned a fear conditioning task. So on the y-axis is somehow the behavior, right? And somehow if the behavior is high, then they really learned the task, right? And that's what you see in the control group. Somehow they learned the behavior, and then on the x-axis you see the days after they learned this, right? The day after the conditioning. And then you see in the control group that with time, of course, they slowly forget, right, this behavior. Good. And then you also see in the lesion group, right, that, and this is where it's interesting, on somehow, and this is a hippocampal lesion group, that on the day 0, right, they had basically, and here's the important point is, when was the lesion made, right? So the day somehow now indicates when the lesion, or these points when the lesion was made. So when you do the lesion very early on, right after the learning, you'll find that this impacts the behavior, right? Quite a lot. You don't see the behavior. Then when you would do the lesion later on, right? For example, after 30 days, the hippocampal lesion, you would see that somehow the memory of this certain, event, of this behavior is not at all affected, right? So this tells you that the brain area, the hippocampus, is important somehow for short-term memory storage up to a couple of days, but not really for long-term memory storage. Good. Okay, good. So to understand this better, I would like to start now diving into the substrates of neuroplasticity, and I would like to start with the network and the systems plasticity. And these are basically how I see the different levels of plasticity. So you could look, at the whole brain and look which brain areas are more active when somebody learns something, and then figure out which brain areas are involved. And this is what we start with. And then you could also look at the next level, which is, I think, is the local brain circuit at the neuron level. Local brain circuit is networks of neurons, right? How do they change their population activity? And obviously the population activity is built up on single neurons, right, that change the activity. And these single neurons, somehow what they change during learning is the synaptic strengths with other neurons. And that's what you then see almost in the synaptic level. And on the synapse, you can dive even more, right? You can look which proteins, which ion channels now really change if the synapse undergoes plasticity. Good. But let's start with the brain area level. And I would like to show you this. I think, model system for studying learning and plasticity classically on this level has been the hippocampus. And here I show you two pictures that depict the hippocampus. First on the left in a rodent, on the right in a human. See, the hippocampus is much smaller in the humans. Humans just have an enormous cortex, right? And then these arrows basically indicate the projection sites of the hippocampus, right? So you see that different types, dorsal, ventral type of hippocampus, have different projection patterns. Yeah. Okay, good. So what does the hippocampus do, right? On which time scale does it act, and which memories does it store? Why do we know this? We know a lot from this person here, Henry Gustav Mollesen. Also, people refer to him as patient HM, very famous guy. What happened to him? He had seizures, and at a very young age, they did a lobectomy, I think is the right name. Lobotomy, and they removed parts of his brain, amygdala, some small parts of the amygdala, a large chunk of the hippocampus, and I think some small parts of the cortex. Basically, this guy had, lost his hippocampus. Then you could, of course, ask, what is he not, what's he now incapable of doing, right? And what they found is basically this, right? He had a normal STM, so a normal short-term memory. So if you would tell him things, you could basically repeat those. He had a normal long-term memory. So if you would ask him, who came visit you last year, right? He could tell you this. He seemed to have a problem in transferring from short-term to long-term memory, right? So if we would learn something new, he could answer, give you answers, but a couple of days or weeks later, he would have forgotten this, right? So he didn't transfer the memory from the short term into the long term, right? And another striking fact that this was only for declarative knowledge, right? So only facts and other things. He was still able to acquire implicit knowledge, such as riding a bike, and he would not forget this. He would, nicely transfer this even into long-term memory. Good. Okay, so the conclusions that kind of the doctors basically draw from this, and there's a very fascinating book from his doctor who kind of accompanied him for like 45 years and studied him. He died in 2008. There's much more cool things that you can learn about this, is that the hippocampus in our brain is not a permanent storage for explicit knowledge. The hippocampus is involved in the consolidation of memory, right, from short term to long term. And then, the other idea is that came out of this here is that somehow the long-term memory consolidation really involves hippocampus, but also cortex. The big idea in the field at the moment is that the memory is moved from hippocampus to cortex, right? And then once this transition has been taking place, you basically can lesion the hippocampus. And that's what this rat experiments that I showed you a couple of slides actually did, right? They lesioned the hippocampus and they don't see an effect on memory storage. Good. Okay. That's basically the big idea behind how memory is consolidated over time, is that first you have some type of ensemble, some type of representation that's learned across hippocampus and cortex. right? If you don't have a hippocampus, you cannot learn this. And then over time, through basically replays, right, through repetition, you will form cortical modules, cortical attractors that somehow are able to represent this memory. And then you don't need the hippocampus anymore, right? Check out this nice review from Jan Born. It's very cool. It's exactly about kind of this idea to push the memory from, you know, short term into long term. Good. Okay. Good. Okay. All right. Good. So I would like to move up from here and talk a little bit about cellular plasticity, and then about the perceptron. And maybe we start with the perceptron. Okay. Good. So this is now, we're on this level, right? We're on the level of local brain circuit and on neural plasticity. Many of you kind of think about this differently, right? They think about, okay, you know, I have a huge deep network, right? And I want to train this, right? And this might, there's studies that have shown that the brain might actually also implement this hierarchy. But I would argue that what we need to understand really first is, what does every single neuron do, right? Which plasticity rules, which learning rules does every neuron follow? And then we might be able to construct a network, use some type of, you know, global connection or some type of architecture to put a network together that actually does what we want. Good. And these single units in this network here are basically, you know, called perceptrons. Good. Okay, good. All right, slides are not changing. Good. All right. So so we move here to the perceptron, and then we go to basically from here. This is like a computational idea of learning, right? What can you do with learning? And then we move to the biological mechanisms of plasticity in a single neuron. Good. So the perceptron was first called McCulloch and Pitt's neuron. And the idea is they basically looked at these beautiful drawings from Ramon y Cajal, and then somehow thought about, okay, what type of computation could this neuron do? The idea that they came up with is that there are inputs into these neurons from other dendrites, right, in the form of electrical signals, depicted here in red, and that these signals propagate forward to the soma. The soma does some type of integration and then sends out the result to the axon. And McCulloch and Pitt is very interesting. They were a biologist and a mathematician, right, that sat together and kind of developed this idea. Good. Okay. Okay, good. Okay. And then the idea is somehow what happens in the soma, right? The idea in the soma is that the soma basically sums up, right, or integrates all these inputs. There can be, of course, positive ones and inhibitory ones, right? And then there's some threshold, some nonlinear threshold. Then once this is crossed, right, then the cell fires an action potential, or also called spike. And this spike is very uniform, right? It's had always the same form, the same height, the same width. So people really think of this as all or none, you know, digital, almost like a digital signal. Good. So how can we kind of formalize this perceptron, right? Okay, good. So this is basically what they came up with, right? This is the idea. And here we have the inputs to the neuron as x1, x2, xn. Then what you do is you multiply with the weight. This is kind of the synapse, right? How big is the synapse? You can think of it as the size of the synapse. And then you sum all these inputs in the soma, and then you push it through a nonlinear threshold function, in this case, a step function. And so what's the difference between the McCulloch and Pitt neuron and the perceptron? So it was called by Frank Rosenblatt a couple of years later in his dissertation. He called it perceptron. And that was then taken up, basically, this naming by the field. The difference that he did is that he said, well, my inputs, right, as well as my output from the neurons cannot only be binary, cannot only be 0 or 1, right? They can take basically all values, right? That was basically the difference between McCulloch and Pitt's neuron and the perceptron. Okay, good. Okay, good. Come on. Okay. So, so the output of the neuron, right, is then very simple. We'll have this threshold function, right? So we define a threshold. Here we can just say somehow that the output is then 0, right? If the summation over the input times the weights is below this threshold. If the output is 1, then we have our sum, right? Our input times the weight is above the threshold. And usually in these neurons, we have a bias, and the bias you can think about as an input that's always 1, right? And then the W0 weight defines how strong this bias is. Good. And the lower, the equation number 2, is just the vector form of this equation. Okay, good. So let's go through an example. What would we get, right? So here we have a weights defined of 0.5 minus 1, and the bias is 1, right? Good. So what would this neuron do, right? This neuron somehow, right? When would it spit out 1? When would it spit out 0? So it would give us a 1, for example, right? If we sum up over all the weights times the bias. And of course, this should be, right? If it's 1, it should be bigger than 0, right? Good. What does this mean, right? If we plug in the weights, that means 0.5 times x1 minus x2 plus 1 should be bigger than 0. We can rearrange this a little bit, and we come to this inequality. x1 is bigger than 2x2 minus 2, right? Now we can exchange the inequality sign with an equal sign, right? And the question for you is, what can this neuron do or implement? Right? Any ideas? So what is this neuron doing? If you think about input patterns, right? There's all types of input patterns, and then for some input patterns, it spits out a 0. For other input patterns, spits out a 1. Yes, classifies. That's correct. Yes, yes. So it's implementing a linear classifier, right? Very simple. And our decision boundary is basically defined, right, by this equal, when I substitute this inequality sign with an equal sign. And then everything below this, right, is exactly the neuron spit. So the perceptron gives us a 1. Everything above this gives us a 0, right? Good. And then, of course, this decision boundary, right, I can somehow, if I wanted to, I can change here. Good. I can change it, right? If I change the weights, then this decision boundary also changes the weights and the biases, right? So now if I have data points, right, I can basically use this neuron to implement a linear classifier, and then I might be lucky that my problem is linear separable or not. But if it is, then the perceptron is actually pretty nice. Good. So how do I train this thing, right? How do we think about learning in the perceptron? And this is the perceptron learning algorithm. It's actually very simple. So I have these two types of stimulus inputs, P and N, right? And I have a label. I want all the P inputs to generate a 1 output. I want all the N inputs to generate a 0 output. I initialize my weights randomly, and then I learn. And so what I do is I pick a random data point, right? A random input, P, for example, and then I look, is my summing up over the weights, is it bigger or smaller than 1, right? If it's bigger than 1, right, that's great. I don't do anything. If it's smaller than 1, or here's smaller than 0, right, then I would need to increase my weight, right, the specific weights. And I'll just, the new weight is the old weight plus x plus the input, right? So I add up the weight vector and the input vector. And the same I do if I put, if I pick an input from N, but my summing over the weights is bigger than 0, then the output of the perceptron is 1, right? That's not what I want. So I subtract basically the input vector from the weight vector. Okay, good. And by this, I somehow converge, right? At some point, and then I have a linear classifier that classifies my data. Okay, good. The question for you here, this is basic. Yeah, this is a basic. Matteo, yeah, somebody said something. Good. Okay, so this is a basic table, and here the question is: can the McCulloch-Pitts neuron or the perceptron, can it implement this function? Right, it's a very simple logical function. Okay, we can in principle draw this, right? So here we have 1 and 1. Here we have my x1, x2, right? So we basically have these data points here, right? All right. Good, right? And we somehow, right, we wanted to spit out, yes, correct, yeah, and we wanted to spit out, right, only give out a 1, right, if somehow for this red data point, right, but we don't want it to give out anything for the other data points, right? So, and as you see, a linear classifier, can have, and there's multiple solutions to it, right? Anything that, separates these two data points, so data point clouds, right, does the job, right? And now it's up to us to use learning and find the weights and the biases to actually, find the classifier that somehow fulfills this. Good. Okay. And the question, which logical function does this perceptron realize? What do we have here? So the -2 is the weights, and then the 3 is the bias, right? Any ideas? None. Yes, correct. Good. It's the non-gate. It was in my slides already. Okay. So this is the check, right? If we have input pattern 0, 0, right, it produces an output of 1. If the input pattern is 0, 1 or 1, 0, it also produces 1, right? But if the input pattern is 1 or 1, then we produce a negative, right? So it's almost the opposite as on the slide before. Yeah. Not the AND is there, not AND gate. Good. So we can, in principle, right, we can implement the OR gate, we can implement the NOT gate. Is there any somehow logical operation that the perceptron cannot implement? You can think about this visually, right? Think about, you want to separate a bunch of data points. Yes, good, correct. Okay. Yeah. Yeah, that's correct. Yes, so the XOR function is a problem, right? See this just very simply from this graph here, right? If you're allowed to draw one line and you want to separate the stars from the circles, right? This is just not possible with one line, right? So the XOR is the classical function that cannot be implemented by the perceptron or by the McCulloch and Pitts neuron. Good. And now, why is this a problem? The problem is because when you look into more real-world data, you want to classify images or audio samples or speech or anything. You're in this highly nonlinear separable data space, right? These data spaces look like this or look even more weird and tricky, right? And you really need more sophisticated classifiers that can basically draw any type of decision boundaries around your data. Do you know what the solution to this problem is? So if the perceptron, what can I do? Can I change the perceptron to, basically learn this type of classifiers? Any ideas? Joining them together. Yes, correct. Yeah. That's the idea, right? I go to deep networks, right? I build networks out of perceptrons. Then I can implement highly sophisticated nonlinear classifiers that, even classify images and everything I want. Yeah, good. Okay, good. This is my summary, and then I think we're up for a break. McCulloch and Pitts neuron, the perceptron implements a linear decision boundary. This is defined by the weights and the biases. We can implement all functions except the XOR. And the perceptron can be trained in a supervised fashion. We need labeled data sets to learn this classifier. Good. And this is the answer to the question, right? Of course, if we then put the perceptron in a deep network, and we train it in a different manner, right? That we basically, or we train it in a similar manner, actually very similar to the backpropagation training, but now we backpropagate the error through the hierarchies, then we can learn much more complex decision boundaries, and that's exactly what these deep networks do and why they are so powerful. Okay, good. So let's make a break here. We have time for taking questions, Matteo, I think, and then I think we resume at 9:15 or something, right?
Yeah. Yeah, I don't think there were any. Okay. So what is the function of the bias? Oliver asks.
The function of the bias. Yeah, it's a good point. So what you want to do is in a deep, for example, if you have a deep network, then you would like to have the different neurons kind of learning different things, right? Imagine you would have two neurons in a deep network that have exactly the same connections and the same bias. Then they would learn exactly the same thing, right? They would be redundant. So what you do is in larger networks, you initialize the weights and the biases randomly. So all neurons are kind of very different. And then some neurons learn a different thing than others, right? It's basically, think of it as a possibility to distinguish neurons and make them more specialized for input A versus input B.
Okay, so we can readjourn. Okay. So we can only find linear classifiers in the brain as single neurons. Also, don't most neurons behave as perceptrons already?
Yeah.
At least those with multiple axon dendritic connections.
That's a super cool question. There is a recent paper from Konrad Kording. I think there's actually multiple. Give me a second. Maybe I'll find it here. I can find it. So what's the idea? The idea is that, of course, a single neuron is somehow much more powerful than just a perceptron, right? Because, and in the Kording paper, they write actually a single neuron is as powerful as a 3-layer neural network, right? Why is that? Because when you think about where do you have, for example, the first nonlinearity in a neuron, right? It's not at the soma. What you'll find is that you, at the first place, right, synaptic inputs come in, are integrated locally in the vicinity of the dendrite, and then a dendritic spike is triggered, right? So that's your first nonlinearity. That is what I think of, might be a perceptron-like structure. Then this result of the dendritic spike is propagated forward to the soma, right? And then again, you have some type of, variables that determine how strong the signal is propagated toward the soma. And then you have a second level of integration, right? And at the soma then is triggered another spike. So I think of a cell, much more as already one biological neuron, right? Integrates multiple nonlinearities and multiple hierarchies probably, right? So it's much more powerful, that's at least the idea in the field at the moment, than a perceptron.
I hope this answers the question. So we have 2 more questions. So can you go to slide 42, please?
42, yeah, sure.
42.
That's this one, right?
Yeah, so the question is, can you explain how you know that at slide 42 it is none?
NAND.
Yes.
Yeah, so,
sorry.
yeah. I mean, you can, in principle, just run the numbers. Where are they? Coming. Good. Here, right? So NAND means, right? So if it's an AND gate, then it would only fire if somehow it would fire, right? It would put out 1 if X1 and X2 are active at the same time, right? Otherwise, it would not, right? So here we have a not AND gate. So it does the opposite, right? It's always active except when X1 and X2 are 0, right? Sorry, it's always active except if X1 and X2 are 1. But when you run the numbers here, right? If X1 and X2 are 1, then you have somehow, -2 plus -2 is -4 plus 3 is -1, which is negative, right? Negative number would not trigger the neuron, it would not spike, so it would put out a 0 here. Right? Otherwise, if one of these things is only 1, right, or none of those, then it would actually because of the high bias, it would spike. It would generate a 1. So it's exactly kind of the opposite or the inverse of the AND gate.
Should specify
Yeah,
3.
activation threshold is 0 here. Yeah.
Sorry?
The activation threshold, I always said it's 0. I always put it to 0. Right?
Exactly. So 3 is the bias in this case. There's just some confusion in the chat.
Yes.
So so this will output.
Yeah,
something
it's not
bigger
the
than
threshold
0,
of, sorry, I said this.
Yeah, I think now it's clear. So there's one more question from a more biological side. 2 more questions. How are the weights determined biologically? The amount of neurotransmitter released by the presynaptic neuron.
Yeah, we'll talk exactly about this in the next half. It is defined by, there's a whole cascade, right? So there's somehow first, you see that the exponential arrives at the synapse, then releases calcium, right? So, and how much calcium it releases depends on a couple of factors. For example, the density of calcium channels. And then this calcium leads to the fact that the vesicles fuse and burst with the synaptic membrane. And the vesicles contain neurotransmitters, and these neurotransmitters are released in the synaptic cleft. And of course, the more vesicles burst, the more neurotransmitter released, the stronger usually, you have the EPSP, so the signal that arrives at the postsynaptic site. But what you also need is, of course, you need a lot of AMPA receptors at the postsynaptic sites, right? That then also could be a bottleneck. And yes, so there's multiple things to that, but we'll have this in a couple of slides. Yeah, we'll discuss this.
And I think there's a last question, which I'll try summarizing because it's quite long. Tell me if I did wrong, if I'm summarizing it wrong. So how do you determine if a specific brain area is sufficient / necessary for something? Yeah.
So necessary is rather simple. You can think about, in the old days, people did lesion studies, right? People did a certain behavior, then lesioned the brain area of interest. Now there's more sophisticated tools like optogenetic. You don't need to lesion it. You can just on the millisecond inhibit this brain area for this task, for this learning, and that is how you would find basically, or how you would investigate necessity. Now, sufficiency is more tricky, right? There, what people usually do is either electrically stimulate this brain area or use optogenetics somehow to stimulate this brain area or the neurons of interest or specific neurons that they think drive the behavior. And if they do this, right, and somehow they stimulate in this brain area certain neuron populations and the behavior is triggered, then you also say it's sufficient to drive the behavior. Yeah, that's this kind of big debate between necessity and sufficiency.
All right, yes, I think we answered that question. If you're okay with it, maybe we can have a 10-minute break. We start at 20, or up to you.
Yeah,
if you want.
Time with me. Yeah.
Do you want a 5 or a 10-minute break?
We can do 10 minutes. It's fine with me.
Okay.
Yeah.
So we start at 20. We had a 10-minute question.
That's good. Then we go into biology again. All right. See you in 10 minutes.
Yes, I'll pause. Yep, done. Good job.
Okay, good. So I would like to continue and then bring in the hippocampus again as a model system for studying learning and plasticity in the brain. And the hippocampus has been very famous for, for explicit, for declarative memory, but also for spatial memory. And this is particularly important when you study mice and rats, when you study rodents. And this picture here on the left basically shows you the activity of a cell, a hippocampal so-called place cell. And the animal here was running around in this squared maze. You see the tracking in black. And whenever it was entering a certain spot, a certain place in this environment, then this cell fired. That's why these cells are kind of known as place cells. And they were discovered by the Mosers, Maybert and Edward, and John O'Keefe Moses also then later on discovered grid cells, and they got the Nobel Prize for this in medicine. Good. So how can we, think about this place cell? We just talked about, the perceptron. And how I think about it is somehow that this place cell also integrates, a lot of different features, right, from this place. And obviously it's not a low-level features that it integrates, it's very high-level features. So what is really important for the rodent, for example, to know that it's in a certain place is the texture of the floor, right? It's really critical to know where it is. The smell is a very crucial cue, the background noise, also the visual input, right? It has been shown that rodents by visual input alone in principle can, know where they are, right? So the idea is that somehow the cells get these different inputs, right? And then the cell tunes like the perceptron to a certain place that, basically represents a certain combination of these features, right? That's how I think about also high-level spatial coding, right? In principle, it's nothing else than, one place integrating a certain set or combination of features. Good. This is what I showed you last time, right? We had this mouse running around this linear track, and then you saw the place cells. The other experiment that people have been doing much before, these linear tracks is called the Morris water maze. Coming up in a second. Good, coming up. Good. Here's the Morris water maze, and the Morris water maze is also a spatial learning task. And in this maze, there's a mouse, as you see here, in a tank. It's swimming around, and it needs to find a hidden platform. Now the water is made milky, so the mouse cannot really see this platform. What it can do is it can look around, right? It can look around in the room, where's the door, where are the lights, and then it roughly knows where it is, and then it can swim to the quadrant or to the area where it thinks the platform is hidden, and then it can swim there and search for it, right? And this experiment shows you on the left that mice can pretty much do this, right? And rats, once they have learned where the platform is, right, in this tank relative to the room, they just search in this quadrant, right? But when you block, and this is, we'll talk about, later what NMDA receptors, what role NMDA receptors play in plasticity. When you block NMDA receptors, specifically in the hippocampus, right? So you interfere with the function of the hippocampus or the plasticity in the hippocampus, then people could show that basically the mouse couldn't learn anymore the spatial representation of where the platform is. Okay, good. So this is then the model system that people use to study on the cellular level, right? So now we know, okay, hippocampus, important for spatial learning. Now we want to understand or study plasticity. Again, we have the hippocampus here. And what people do is they take the mouse brain and they cut it in, a lot of, it's called coronal slices. Think of it as cross sections, right? And one of these cross sections you see here. And because of the cellular structure, the cells actually survive this slicing process. And then you can come with electrodes and patch pipettes and other things, and you can study this whole system, right? It's called in vitro, right? Because it's not in vivo anymore. And you can still study, the excitation of cells and plasticity. And the pathway of the hippocampus is in principle very simple. Information comes in from the entorhinal cortex. So the perforant path, that's what you see here on the right, goes to the granule cells from the dentate gyrus, from there to the CA3. And then the most studied pathway in the hippocampus is really, I would say, the CA3 to CA1. And that's what we also talk about, this, the Schaffer collateral pathway. Good. Now what you can do is, right, because the system is so nicely classified, you can in principle put here stimulation electrodes on different inputs, right, on different Schaffer collateral inputs, stimulate inputs to our CA1 pyramidal cell repeatedly, and then look, how does the response, how does the voltage deflection in the postsynaptic cell change? And that would give you an idea of, under what type of condition the cell undergoes plasticity, which synapses are increasing, which synapses are decreasing. Good. And what people find is basically two a couple of different mechanisms. So the first mechanisms we talked about, different time scales of plasticity, is called paired pulse facilitation, and this is a short-term plasticity mechanism. What you see is that if you give two stimuli, and this could be, for example, just one pipette, one stimulation pipette, and you stimulate 2 times, right? Or you have 2 and you stimulate shortly after each other, then you see that when the stimulation comes very short or arrives very short in time, and the trace you see here is measured in the postsynaptic cell, right? That it actually overlaps and sums up, versus if you then go out and you space it more than 25 milliseconds or something, right? Then you see there's almost no summation of these incoming inputs, synaptic inputs, right? Why is this important? Because remember, we have a nonlinearity in the cell, right? Like in the perceptron, right? So the peak amplitude here is really crucial because it could lead to spiking or non-spiking. Good. Anybody have an idea what the mechanism for this is in the synapse? Okay, good. I'll resolve it. So the idea here is that you still have residual calcium, right, in the synapse. And this calcium, usually it needs somewhat around 30 to 50 milliseconds until it's really pumped, actively pumped out of the synapse, right? And calcium obviously also is, depolarizing. And because of that, you have some type of potentiation. It's building up, right? Yes. And we said, the more calcium, the more neurotransmitter release, and the stronger somehow, the next stimulus or the next EPSPs. Okay, good. So the very classic experiment is something like this, right? Where you in principle start, and I'll just try to explain this, from minute -15 to 0, what you do is you give individual pulses, right? Individual stimulations. And then the y-axis, it's written EPSP field rising slope. What is this? This is something like similar to the EPSP amplitude. It gives you an indication about how strong is the signal, right, that is coming in at the postsynaptic neuron. How strong is the voltage deflection, right? And of course, if this goes up, right, or increases, then we would think about a potentiation, a long-term potentiation. The synapse gets bigger. If it decreases, then somehow we would talk about a depression. What you see is when you do this, right, you give a pulse here and then, you make a break between them, then nothing happens. And then at time point 0, what the experimenters did here, they give a tetanic pulse. What's a tetanic pulse? It's a very strong repeated stimulation, let's say 10 stimulations at 100 hertz or something. What you then see is these PTP, these post-tetanic potentiation. We just talked about it, right? These short-term plasticity mechanisms due to residual calcium. This somehow breaks down, of course, if you stop the tetanic potentiation, right? At some point, the calcium is pumped out, and then you would give your, single stimuli again. And then what you see is that after this strong tetanic stimulation, the EPSP, so the input into, or the voltage signal in the postsynaptic cell has potentiated. It's now much, much stronger than before, and this lasts over 60 minutes, right? So what does this mean effectively? It means our synapse has grown, right? There's now more AMPA receptors, more vesicles, and so on and so on. I'll come to this in a second. Good. So this is long-term potentiation, and this can be elicited, as I said, if the tetanic stimulation or the tetanus stimulus is very high frequent, right? So if this is between 50 and 200 hertz, people find that the cell undergoes L2P, right? Long-term potentiation. The synapse goes bigger. And again, here you have the slope of the EPSP. Sometimes it's measured by the amplitude of the EPSP. EPSP is excitatory postsynaptic potential that you measure in the postsynaptic neuron. Good. Now you could also change this protocol a little bit to introduce LTD, long-term depression, right? So how would we do this? A very simple way to do is you simply change the theta stimulus, or the theta stimulation frequency. If you choose a range from 1 to 10 hertz, so low frequency, then what you will actually find is a depression, right? So what people did here, you see the EPSC amplitude is the excitatory the postsynaptic current, which gets smaller and smaller, meaning also the voltage deflection becomes smaller if you do this low frequency tetanic stimulation, right? Good. So there seems to be some type of boundary, right, of stimulation frequency where the neuron really switches, right, from LTD towards LTP. And we'll talk about in a second, what might be the underlying mechanism that mediates this. Okay, good. This is a brief summary. So we talked about that LTP is frequency dependent, right? It's long term. It means or it involves mechanisms that strengthen the synapse, and it follows this tetanic stimulation. It lasts from 30 minutes, can even last for a couple of hours. And these mechanisms do require protein synthesis. We'll talk in a couple of slides what actually happens, right? And then we have LTD. It's kind of same, also involves protein synthesis. It's also occurs after this tetanic stimulation, but low frequency. And obviously, and this is 2B, right? Think about it, when you have a neural network, you cannot only potentiate all the time neurons, right? There need to be some type of compensatory mechanisms. You need always somehow potentiation and depression, both working together to reshape information coding in a network. That's also with my kind of recent paper, I've basically argued very strongly that memory formation always needs LTP and LTD, and we actually see that in the brain, right? So some cells start responding stronger to a stimulus versus others start to respond weaker. Good, so it's the reshaping, right? A shifting of the population response. Good. And then we'll talk about this at the very end. Spike time independent plasticity is thought to arise from the same set of mechanisms as LTP and LTD. One can actually connect this. Good. Okay, so let's talk about the last thing, the synaptic plasticity and the Hebbian synapse. And here we are at the level of synapse and molecules, so the last stage for today. Okay, so I would like, before kind of showing you how synaptic plasticity works, give you a short recap of what happens during synaptic transmission. What's the idea? So the idea is that first, right, you'll have an action potential arriving at the presynapse. At pre. Then what happens? This action potential causes a voltage deflection in the synapse. Then you have voltage-gated calcium channels, right, that introduce a calcium influx, right? So voltage-gated calcium channels have caused a calcium influx. Then this calcium, this high calcium concentration leads to vesicle bursting, fusing and bursting, right? What you see here. And the vesicles contain the neurotransmitter glutamate that then enters or crosses the synaptic cleft, right? So this leads to vesicle bursting, which then leads to neurotransmitter release. Good. So this neurotransmitter then goes, and the synaptic cleft is really, really tiny, right? So it crosses the synaptic clefts, and then it binds on the other side, on the postsynaptic side, to AMPA receptors. And these AMPA receptors are basically ion channels that are, in this case, selective for sodium, right? So the lead and sodium on the postsynaptic sides, which in turn then introduces a voltage deflection on the postsynaptic side, right? So, and neurotransmitter, right, in this case, glutamate binds to the AMPA receptors, and then we have 6 post voltage deflection. And this postsynaptic voltage deflection is basically your EPSP, right? So this basically shows you that it's almost a cascade, right? And there's multiple things that could be a bottleneck, right? Starting from the calcium channel density to the amount of neurotransmitters to the number of AMPA channels. And it's important that if you want to strengthen synaptic plasticity, you basically work on multiple of these, right? Good. Of course, we also have other neurotransmitters, GABA, which are inhibitory and so on, but we don't, talk so much about these here. Okay, and this is just a little, does it actually work, this video here? Yeah, there's a little video. You see the vesicles fusing, neurotransmitter crossing, and then you have the voltage deflection on the other side. The voltage-gated calcium channel. Yeah, good.
So before you go forward, I think there are two questions that deserve attention. The first one, what exactly does the calcium do that promotes vesicle movement and neurotransmitter release? I feel like for the past lectures, most lecturers have avoided explaining it. Probably don't want to go through in a rabbit hole.
So what is the mechanism that when you have a high calcium concentration leads to vesicle bursting? Is that the question?
I think that's the question, yes.
Yeah. I must say, I don't know. When you looked it up. Good question.
And then I think another question, if the action potential is strong depolarization, what encourages the positive calcium ions to flow inwards? Does it happen shortly after the action potential during hyperpolarization?
Yeah, so important for the calcium ions to flow inwards is the gradient of the calcium concentration, right? And we know that inside the cell, we have about 10,000 times lower calcium concentration than outside of the cell, right? So calcium, if you have a channel, and the channel is not actively pushing calcium into the cell, it's just opening and then letting selectively calcium through, right? Then this concentration gradient basically and establishes a force that pushes the calcium inside the cell.
I think we're all caught up.
Okay, good. So let's continue here. Here on the left, what you basically see is on the bottom, right, is an EPSP, and here you can think about, this is our voltage here, right? Then you have the amplitude of this EPSP, and this amplitude is usually an indicator of how strong or how big is my synapse. So when we look at the synapse, and here we have the pre, right, and here we have the post, then what we see is in the normal state, right, we have a certain amount of vesicles, a certain amount of AMPA receptors. After LTP, after long-term potentiation, what we see in the synapse is more vesicles filled with more neurotransmitter, more glutamate. On the other hand, you see here more AMPA channels in the membrane, right, which makes sense, which then somehow, right, lead to a larger voltage and calcium influx also in the postsynaptic neuron. And this, if you measure this, right, would be, if you measure this as a current, as a voltage deflection, would lead to a higher somehow EPSP amplitude. Okay. And this has also been observed experimentally. I have to say that the number of, what people found is that the number of AMPA receptors in the cell membrane and the relating number of neurotransmitters or vesicles linearly seem to linear scale with the size of the synapse, right? So the idea is the bigger the synapse, the stronger it is, and the bigger somehow is this amplitude, the EPSP amplitude that you measure. And this fact has actually been experimentally verified. On the right, you see a 2-photon imaging experiment in vivo where somehow the mouse learned new things and underwent plasticity. For example, if you look at this spine here, right, you'll see that, through learning, it grows, it becomes bigger, right? And obviously there's kind of multiple spines here that get bigger. And then if you measure the connection between two individual neurons, then you find that also kind of the EPSP size here, right, increases, and it's proportional to the increase in the synaptic size. Yeah. What you have to watch out for is obviously a cell usually doesn't make just one connection with another cell. There's usually a couple of synapses, and what you find is that usually all of them, increase in size or all of them decrease in size. Yeah. Okay, good. All right. Okay, so we talked about synaptic density changes, right? This is kind of on the long term. It's like long-term potentiation or long-term depression. Short-term facilitation or short-term depression would be the calcium levels that sum up. What you often read in the literature when people talk about plasticity is that the AMPA NMDA ratio changes. So there's of course more ion channels in the synaptic in the synapse than AMPA, right? There's also NMDA. NMDA is an ion channel that also binds to glutamate, then opens, but it's not selective only to sodium. It lets to sodium and calcium, right? Now, when a cell undergoes plasticity or a synapse, then what you see is that the number of NMDA channels or NMDA receptors usually stays constant versus the number of AMPA receptors stays, right? So a very good measure of looking how much, how strong or how much the synaptic strength increased is to actually look at this ratio. Good. And then what you see on the very long term is that the number of spines changes, right? And this image here on the bottom basically shows you this. Good. To change my somehow Zoom is, oh dear. Good. Coming? No. Okay, good. So what you see is that at the very beginning, right, on the very left, we have the control case. Then after LTP, relatively short term, we see that new AMPA receptors that are basically flooring around in the synapse, recycled, are integrated into the membrane. Then somehow the synapse gets stronger, right? There's more AMPA receptors, there's more glutamate. At some point, what will happen, you will see structural changes and that the spines obviously cannot grow and grow. So the spines will then divide and become two independent spines. Obviously, these two independent spines are from the same neuronal connections, right? You could basically sum up their effects to specify the connection strengths between these two neurons. And at the very, on the very long time scale, what you'll see is that even on the presynaptic side, you see individual boutons, right, that have individual neurotransmitter release. So these are very long-term changes that you observe. Okay, good. So I would like to come back to this NMDA receptor and what the role of the NMDA receptor really is in synaptic plasticity. So as I said, we have NMDA receptors and AMPA receptors in the postsynaptic membrane. And here people performed an experiment where they did a long-term depression protocol, right? We talked about it, 1 Hertz stimulation, low frequency tetanic stimulation. And in the control case, you would see that the EPSP gets smaller, the EPSP amplitude, right? So that means the connection between the cells has decreased. Now, if you apply APV, APV is an NMDA receptor blocker, then you see this effect is partially blocked, right? And it's always partially in this pharmacological experiments because these APV compounds, they'll never block all NMDA receptors. It's like a certain percentage of NMDA receptors are blocked. Now, why is this happening, right? What's so crucial about NMDA receptors in this whole thing? And the main reason why people think NMDA plays a major role in controlling plasticity is that it also lets in calcium, right? And that the calcium levels are really the determining factor to either introduce LTP or LTD. And this is how people think about plasticity and how it depends on calcium. So first of all, yes, calcium flows in through the NMDA receptor, right? And the NMDA receptor is activated by glutamate. And then in the synapse, in the neuron, right, this calcium binds to protein kinases. And there's different protein kinases either for low calcium levels. And you see, if you look at the plot on the very left, right, the idea is that, when the cell is not active, or the synapse, you have a certain baseline level, baseline calcium concentration. Then you have a little bit of input or a low frequency input, and basically elicits a little bit elevated calcium level. And then at this calcium concentration, you'll have calcium kinase 3 and calcineurin becoming activated. And these calcium kinases are pretty fascinating. They only bind when the calcium concentration is at a certain level, right? At a certain low level. If the calcium concentration goes higher, they wouldn't bind to calcium anymore. If the calcium concentration is baseline, they also wouldn't bind anymore, right? So if you have this low calcium concentration here in red, then you have chem kinase 3 and calcineurin activated. If you then have stronger inputs, for example, high frequency stimulation, then the idea is that you have chem kinase 2 and chem kinase 4 activated, and that these somehow fuel into signaling, intracellular signaling cascades in a cell that end up with LTP, right? So in principle, you can now map this stimulation frequency idea, right, of low and high frequency stimulation onto calcium levels, where I say, well, low frequency stimulation just elicits low calcium levels. I can even map it to a certain intracellular plasticity pathway, and high frequency stimulation would somehow activate chem kinase 2 and 4 and would fuel into a different intracellular pathway that then leads to LTP. Good. Okay, and this is basically just to show you that this whole, very simple sketch that I had before, just chem kinases, right, and calcineurin, is much more complex. There is a huge machinery in the cell that also interacts with each other, right, that determines at the final end if the cell undergoes LTP or LTP. There's also metabotropic glutamate receptors, right, that can bind to glutamate and then somehow down the line can affect or reverse the effects of chem kinase 2 or 4 or 3. So it's much more complex, but just for simplicity, I think the main factors are really chem kinase 3, calcineurin, and chem kinase 2 and 4. Good, just that you know that it's more complex. Okay, good. So what about...
Sorry,
yeah.
About spines, about dendritic spines. So can we assume that spines formed by dividing have the same function as the ones they originated from? Or can the function change?
No, the idea is that, when two cells are connected, right, that all the spines have basically the same function. So when the presynaptic cell is active, right, you would sum up over all these spines to get an idea of the connection strengths between the two neurons, right? And if the postsynaptic or if they undergo LTP, then all the spines would potentiate. If they undergo LTT, all the spines would depress. Yeah. Usually, you find that cells are connected relatively locally. So when an axon passes a dendrite, you find sometimes 4 or 5 contacts that are relatively local, right? It would not usually connect then to another far out branch or something. Yeah. Yeah, that also explains the kind of that all these synapses usually have a similar function. Yeah. Okay. So what are the characteristics of LTD and LTP in terms of when do synapses potentiate and when not? So the idea is that if I have a normal synaptic transmission, I have just a weak synaptic input, I have no LTP, right? Then if I have cooperativity, right? So multiple synapses being active, then somehow I increase the calcium levels, right? I have a strong input into the neuron, my neuron spikes, and then I undergo LTP, long-term potentiation. And then I increase the synaptic strength for all of these synapses. What I can also have is the fact what people call associativity, that I have one synaptic input that's very, very strong, that's driving the cell, right, leading to a lot of calcium influx, and one weak input that arrives at the same time. And the weak input usually also is potentiated. Good. And this LTP and LTD mechanisms are synapse specific. So that means if a synapse is not active, right, then it doesn't potentiate or depress. It doesn't do anything, right? It needs to be active. somehow to participate in the learning process. Okay, good. And that's the idea of Donald Hebb. To kind of give a bit background, Donald Hebb is a physiologist, and in the 1950s, he was looking at the behavior experiments, the Pavlovian condition experiments from Ian Pavlov. And Pavlov had done these at the beginning of the century and basically came up with the idea, right, that animals can learn this association between a bell, right, ring a bell, and then, getting a reward by looking at these dogs. And Donald Hebb tried to find a neural substrate on the neuronal level, right, that could explain this associative behavior. And that's why he came up with this Hebb idea, right, cells that fire together, wire together. So again, you have these two cases. You have specificity. The synapse is somehow strengthened, right, if you have a strong input. But if the cell has no input, right, then it's not strengthened. If both cells are active, you have associativity in that the weaker pathway also gets strengthened. And the idea you see depicted in this coffee pictures here is very simple, right? If I see somehow a coffee mug, I see it, I smell it, I maybe feel it, right? Then I associate all of these features, right, for the coffee. So if I want to have a coffee cell, right, a coffee mug cell, I need to associate all these features. And for that, I might have stronger and weaker inputs, and associativity and specificity would allow me to do that, to learn. Good. Okay, this is Hebb's idea again. This led to 1949 to Hebb's postulate. The simple idea is, this is basically quoted from the paper, when an axon of cell A repeatedly or persistently takes part in firing cell B, then A's efficiency as one of the cells firing B is increased. Simply, you may have heard the phrase firing together, wiring together. There's the basic idea, but here is some more, a little bit more specific in that Hebb's postulate is local in time and space, right? So the cell needs to fire together, and it's somehow causal, right? So one cell drives the other. The update rule here is basically the firing rate of the neuron A times the firing rate of the neuron B, right? If they fire together, right? Yes. Hebb, in his postulate, did not specifically say, he says, yes, there's probably, no matter what, there's some direction, some causality, but he didn't say which cell has to fire first. Good. Okay. One experimental verification of this Hebb's theory has been then published later by Mu-Ming Pu and colleagues. And this is called, you may have heard about it, spike timing dependent plasticity, STDP. What's the idea? What have people found here is when you patch, or when you measure from two neurons here, right, and you excite the first neuron, the presynaptic neuron, and then the second, the postsynaptic neuron spikes, then you can measure a difference between, the presynaptic input, the presynaptic spike, and the postsynaptic spike. And if this difference is positive, right, then this means the presynaptic neuron has fired first, as you see here in the little sketch, and then the postsynaptic, and this is lead to a postsynaptic spike. Then you have a positive increase in the weight, right? Our synapses potentiate. If you have a negative time difference, then we depress, right? Because then, our presynaptic neuron could not have led to a spike in our postsynaptic neuron. That's the idea. And on the right, this little inset, this is basically kind of, if you fit this data, it would look like this curve, right? So the closer they are together, how is delta V measured? Yes, so in principle is the size of the synapse. What you measure effectively is the amplitude of the EPSP, right? So if that grows over time, then this timing has led to an increase in delta W. Good. You can measure it, for example, as it's a percent in change of EPSP. Good. Okay. Yes. And then if you flip this, of course, right? If the presynaptic neuron fires after the postsynaptic neuron, then you have LTD. Okay. Good. And then this is my last slide. And then I have a little quiz for you. just to mention that neuromodulators, right, such as dopamine, norepinephrine, serotonin, and others can really affect this curve, right? So here you see the classical paper from B and Pu and hippocampus, where they basically kind of beautifully measured this STDB curve. Now, when you release dopamine, right, onto the slice, then what you see is that this curve actually changes. And that's what you see on the bottom in green. What you see here is that you see again the change in the synaptic change in percent, right, which is basically is the same as the change in weight, right, in the previous slide. And you can simply measure this by looking at the difference in the EPSP amplitude. Yeah. What you see here is that, you know, especially the positive or the negative side has flipped, right? So dopamine and others seem to have an effect that this timing, right, is not so relevant anymore. Just the pre and the postsynaptic neuron, right, the order is not relevant. If they fire somehow together, right, disrespect this of the order, you have a strong change in the weight, versus if they fire, you know, apart, then you would not change the connection between them. Good. And this is very cool. Think about, you know, the role of dopamine as a reward signal, right? Think about the role of norepinephrine and other neuromodulators as signaling reward, pain, all these, you know, global arrow signals, if you would call them, right? They really affect how cells on the single cell level and how synapses grow and how they evaluate, right, the timing information between pre and postsynaptic impulse. Good. All right, so we have, I think, a couple of minutes left. So I have this little quiz that I thought we could go through. So I'll just go through and then to the questions, and then let's see if you know the answer. Maybe too simple. A receptor critically involved in LTP is one, NMDA, AMPA, GABA B, or CAM kinase 2. Yeah. Yeah, correct. Number one. Okay, good. CAM kinase 2, a calcium-dependent protein kinase. Which answer is correct? Binds to calcium at a specific high concentration and triggers LTD. Binds to sodium at high calcium concentration and triggers LTP. Binds to calcium at high concentrations, calcium concentration, and leads to LTP. Or binds to calcium at low calcium concentration and leads to LTP. Yep, number 3 is correct. You got it. Okay, good. NMDA receptors work as a coincidence detector in LTP. The two signals that must be present are presynaptic signal followed by calcium influx, a presynaptic signal followed by a postsynaptic signal, a postsynaptic signal followed by presynaptic inputs, or activity signals in at least 2 or more synapses. Right, guys, yeah. Think about Hebb, right? Donald Hebb. What did he postulate? What needs to be first? Number 2. Yes, correct. Presynaptic followed by postsynaptic signal. Okay, good. All right, 2 more questions. The importance of NMDA receptors for LTP can be seen in a behavioral experiment with hippocampus knockout mice. This is the experiment I showed you in the Morris water maze. What do we observe there? Mutant mice learn significantly slower. They don't learn at all. After a few hours, right? Think about hippocampus and its time involvement. After a few hours, mutants don't remember. After a few days, mutants don't remember. That's tricky. Think about also this, you know, this lesion experiment, right? When did the lesion have a big effect? Biggest effect. Yeah. And these are knockout mice, right? It's maybe a tricky thing. It's not a lesion. They, grow up and they don't have NMDA receptors in the hippocampus. Okay. Good. So they don't learn, right? If they are knockout mouse. Good. Okay, good. It's different from a lesion experiment. Okay, good. Last question. During the late phase of LTP, or the very late phase, right, the number of synapses increases, number of synapses decreases, there are less transmission failures between synapses, and the increase of synapses can be measured. Okay, yes, it's one. Correctly, yeah. Yeah, coming back to the upper question. So these knockout mice, right, they basically, they grew up and they don't have expressed NMDA receptors in the hippocampus, so they either, depending on how good the knockout is, don't learn at all, or they learn super, super slow. Yeah, that's different from the lesion experiment, right, that I showed you, where basically the mouse brain hippocampus are intact. They form a memory and then you lesion. And then you figure out what type of effect does this lesion have shortly after the learning or long after the learning.
So everyone always asks for exam questions or mock questions, whatever. These sort of quizzes can be seen as mock exam questions for everyone listening.
Yeah, more. The exam questions are would be more. The type would be different. It would be more one question about a topic with a couple of sub questions, right? This is just checking your knowledge. Yeah.
They're quite similar, let's say. I mean,
no, I think they're actually different in that, exam questions are structured that you have one topic, let's say LTP, and then you have a couple of sub questions, right? So one could think of, basically combining 2 or 3 of these questions to make one topic, and then this could be one exam questions.
No, I don't think so. Carla?
And this look like, yeah, this look like no.
Like it's 50 multiple choice questions on all the topics of the course. It's
Okay, yeah, then it was different last time maybe, but was it multiple choice? Are you sure?
Yeah. It's
okay.
Types of questions, multiple choice and true or false. And so
Okay.
these could be
okay,
example.
good, then I take it back.
No, sorry. So there were a few questions before the pool. Let's see if I go back. Okay, so if strong stimulus were to reach the postsynaptic neuron, could we also have associativity? Or the first weak input would be in hyphens lost?
The first weak input. Yeah, so usually the idea is that if the strong input arrives and the cell spikes, right, then the timing for the weak input is crucial. If it's before the strong input, you'll have potentiation. If it's after the weak input, you'll have depression. Now, you could easily, if there is, right, in addition, dopamine release, which we saw here, right, then the timing doesn't matter so much, right? Then it's really just important that the weak input arrives roughly around the time where the strong input comes in and the cell then spikes.
Then what happens at time delta equals 0 milliseconds?
That's a good question. Yes, it's exactly 0. Yeah. Yeah. I mean, there are, as you see from the data points, right? There is one data point that is exactly 0, right? Yeah. I guess it's very tricky. I guess it actually happens that it is 0. You don't have any change in the synapse, but that rarely happens because you're just a little bit off to the right or to the left, and you are have a very strong depression or pronunciation.
And then can you go to slide 73?
73. This one.
Yes, so they asked, can you repeat what the y-axis means in figure 70? But I think you just went over it.
Yeah,
Yeah.
it's the change in the weight, right? And you basically measure this. I think here is also measured in percent of, so you measure the EPSP amplitude, right? So you measure something like this here, right? The amplitude of the EPSP. And then this, if you have potentiation, it grows, and you can measure what percentage does it grow, right? And the percentage then you basically define as change in the weight. Right? So your weight has increased by 50% or something, as measured by the relative change in the EPSP amplitude. And that's also kind of the same measure you see here, right? The synaptic change in percent, strength of the synapse.
So we have two more questions. Does long-term potentiation simply mean delta W greater than 0? Or is there more to it?
It means delta W bigger 0, but it has a temporal component, right? It means that, this delta W0 basically holds for a couple of minutes to maybe an hour or something. Yeah.
And then changing topic a little bit. So going to dopamine. So
Yeah.
dopamine switched learning from causal to correlative. Why does
Yes,
sense
pretty
for a
cool.
reward signal?
Sorry? Didn't get. Why?
How does this make sense for a reward signal?
Yeah. People don't know. I find this, so check out the paper from Frimeau and Gerstner, the review about neo-Hebbian learning, where they basically say, look, Hebbian, is always in principle correlative, right? You could have a positive or negative correlation in time. And the problem with this is that it doesn't take into account any global error or any reward, right? So what they postulate, and this is kind of, I think, Wolfram Gerstner's postulating this for 10 years now, is that there is an additional factor, a global reward factor that, also determines how the timing is evaluated of pre and post, right? And that somehow, this then allows you to reshape the STDB function as you see it here in a way to account for global errors. That's the idea. How that, how you would actually create or train a deep network for reinforcement learning or something, right, where you use dopamine as an error signal to modulate this STDB function is not clear. I don't think anybody has really successfully done this.
Another change of topic. What is the effect of the active protein kinase in the neuron? Why does it cause LTP?
So the effect is at the very end, there's a whole signaling cascade, right? It goes through, it activates PKA and MAP2 pathways and so on and so on. And at the very end, it leads to an increased expression of AMPA receptors at the soma, right? So the cell produces more AMPA receptors. These are transported to the synapses and then built into the synaptic membrane. Yeah, that's what these CAM kinase 2 and 4 pathways lead to. And so think about it, you have a cascade, some ribosomes that lead to the multiplication or expression of AMPA receptors, and you can speed up this process, right? This would be the LTP pathway, and you can slow down this process. Then less and less AMPA receptors would be produced, right? So the synapses would then somehow integrate or recycle AMPA receptors and disintegrate them from the membrane. Yeah. Why they're called CAM kinase 2? This is calmodulin kinase. It's I think, yeah, I don't actually know anything. I'm looking at why they're called like this.
Yeah, so I think
calmodulin is the part that binds to calcium, right? And kinase is the part that then actively triggers the signaling cascading and stuff.
Yep. So I think these are all the questions. I hope I didn't miss any. I don't think so. But
Okay, good.
yeah, so thanks a bunch for these 2 lectures.
Yeah.
For
start with the exercise, I think, right?
exactly. So I was going to suggest that we meet at 10:20, like before, and then we can go through the solutions of last week's exercises. And I already put online this week's exercises. And yeah, should be good.
All right, good. Thanks a lot to everybody. Have a good day. Bye-bye.
Thanks, man. Oh. Whatever.
Okay.
Okay. Moving on. So then, now that we know what the single neurons do, we can, and now we have a new raster plot. We can figure out the trajectory in 2D space of this new trial. And so, for example, we have, as I said before, B and E that are movement on-off cells. We have our head direction cells. We have our grid neurons. And so we can kind of construct this 2D, this this new path, which is a 2D map. And so yeah, I mean, you can see in time, at time 2, E starts firing. E is a movement on cell, and so we can make it move from E. Okay, any more questions? Okay, and so then the third exercise was a lot more theoretical exercise based on Poisson spike trains. And so for this, I want to go read the exercise with you guys because it's a little more complicated. So spike trains are simply a sequence of events and are often represented in a raster plot. A spike train is a sequence of events that happens with particular statistics. You can get to know some of these statistics in this exercise. So we have 2 assumptions about our spike train: that there is no dependence on the timing of an event on the on preceding events. So the next spike will be independent of previous spikes, and that the firing rate is constant. So we can expect that R delta T spikes in a short interval of delta T. So now we have our time vector. So this is our normal time, and we discretize time in small bins. So every bin normally is, we can say, 0.1 milliseconds. And doing so, we are absolutely sure, and now we will show, that no 2 spikes will ever fall in the same delta T. This is just a way to model biological spike trains. And so the first question is, for a given T and delta T small, compute the probability that there are exactly n spikes in the trial of duration T with bin length delta T. And so for this exercise, we need to consider a few things. The first thing is we need to see the number of ways we can put n spikes into m bins, right? And so this first term is just a binomial of mn. Then we need to look at the probability of n spikes occurring in n specific time bins. And so, so we know that the probability of a spike occurring in a specific time bin is R delta T. And so if we want n spikes, we just multiply that, we elevate that to the n. And then we also want the remaining m minus n time bins. So m is our global number of bins, n is the number of time bins that are occupied by spikes. We want all of those not to have a spike, right? And so then we also have to multiply by this other factor. And I just want to shout out to the Diane Abbott book. So there's this book called the Dynamo Theoretical Neuroscience, and this is kind of the foundation of all of theoretical computational neuroscience. It is freely available online, and you can derive all of this, and it's very nicely explained, much better than I will ever be able to explain it at page 26. You can there's the whole explanation here. I can post this chapter online if you guys want. I highly recommend it in case you're interested. Keep in mind, this exercise is not mandatory.
Can you show the slide in full screen?
Just if it's full screen, then I cannot go to the exercises. So I'll read this and then I'll go back. So using the properties of the Poisson distribution, we can confirm that the expected number of spikes in the interval T is RT. The variance of the spike count in this interval T is also RT. So then they ask, the Fano factor is defined as the ratio of the variance and mean of the spike count. Which value does it have for Poisson spike trains? And so, okay, I can make it big. And so we know that in this case, the Fano factor is 1, but we also know that we know from statistics that a Poisson spike train, the mean and the standard deviation are the same thing. And so this sort of confirms the fact that Poisson spike trains are a good way of modeling it, because we arrive at the same conclusion. And so finally, number 3, the probability density of time intervals between spikes is called interspike interval. And it's a useful statistics for characterizing spike trains. Suppose that a spike occurs at time ti. For small delta t, what is the probability p that the next spike occurs somewhere in the interval ti plus tau and ti plus tau plus delta t? What probability density p of tau do you get? And plugging in the numbers, we get that the probability of not firing a spike for a period tau times the probability of generate a spike within a very small interval. And so plugging in the numbers, we get this. Sorry.
I don't understand the purpose of these bins.
Okay, so the bins, so okay, let's take a step back. I'm sorry. We have, look at me, not at the slides. So we have our time t. So we're making an experiment that lasts 1 minute, 2 minutes, 3 minutes, doesn't really matter. And so I wanna have a mathematical tool to be able to model the spikes that I will record. So I will record the spikes and then I will have all my, hello? Oh, sorry, I heard an echo. I will have all my spike times. And then I wonder, okay, how can I model this? And so one way of doing it is I take my time and I divide it in very small time intervals. And then I check for every time interval if only one spike has occurred. So let's say I have a recording of 1 minute, and let's take I take a bin delta t of 5 seconds. And so the probability of having multiple spikes in 5 seconds is pretty high. And this would not be a good way of modeling such data, because we cannot afford to lose the fact that a spike occurred. So then I can pick a delta t of 0.0001 milliseconds, 1 milliseconds. And so then the probability of having multiple spikes in the same time bin is basically 0, because you know that there's a refractory period, and so the neuron physically cannot go above a certain firing rate. But this is also kind of computationally stupid, if you want, because most of these delta t's will not have a spike. And so you are kind of, it's an overkill. And so then you can say, oh, the refractory period is around 1 millisecond. So it's a safe assumption that if I take a dt of 1 millisecond, then there will only be one spike per dt. And to be safe, maybe you can make it half a millisecond, but that's how you get to a dt length. Carla?
No.
Questions? No questions?
No, there is another one.
I don't know if
I find
how I explained it is clear.
wait, I find these transitions from discrete to continuous distribution somehow strange. Wouldn't a geometric distribution maybe be more appropriate than an exponential distribution? Oh, but this is
So going from continuous to discrete is something that we have to do if we want to be able to model life, right? And now the question is, okay, what is the most
so annoying.
Can you mute her? Okay. I muted her. So this is a transition that we have to do. And so now a question arises, what is the best way of doing this discretization? If you're interested in this, and this is very optional, it's not in the scope of this this program or in this course, you can read that chapter of the Diane Abbott in which they explain very nicely how the Poisson distribution is the correct distribution for this approximation. I'm sure you can find kind of any distribution, but you kind of also have to look at your data, your input data, the data you're recording to see if it fits or not.
yeah, there's
Okay, so I will try putting some stuff on place cells and grid cells, on the solution exercise, because while the math behind a Poisson distribution is optional, place cells and grid cells are quite important. And if really anyone wants, I can also put the theoretical neuroscience book online. I can actually put it. Okay. What's up? Oh, nothing. If there are no more questions, then I think, yeah, that's it for today.
I would suggest that we wait a
Yeah.
little bit, and what we can do is that because we forgot to record, we can just repeat the first part at the beginning right now, just so it's in the recording.
Okay.
Even if they leave,
Sounds
we just
fair.
leave it the same recording. For what should be the displacement between t 3.5 to t equals to 5? I mean, do we compare with the figure 2 to get the velocity of the creature? Since I noticed there are two density of c, which